Hello, my name is Tanya Diaz-Gonzalez. I am a sophomore biology major, and today I'm going to be talking about pesticides and pathogenic contributions to colony collapse disorder in Apis mellifera. So before I begin, I want to briefly talk about how I selected this topic. So years ago, I remember seeing that honeybees were an endangered species. And while doing my research to do my talks, I saw that there were a lot of threats to these species, and one of them that caught my eye was colony collapse disorder. So it seemed like the most detrimental thing towards honeybees. So I wanted to learn more about it. And it is a very interesting topic because it is a mystery that has been going on for almost 2 decades. But there have been a lot of important research that has been done in order to help the species and their survival. So that is how I selected my topic. And without further ado, we can get started. So this is a brief outline on what I'm going to talk about today. So first, I'm going to talk about the background, what Apis mellifera is, what colony collapse disorder is, and some contributing factors to colony collapse disorder. Then I'm going to go through the experimentation, the approach, methods, and the results. And finally, I'm going to end with a takeaway, the conclusion, and what the study means for the future of honeybees. Apis mellifera. What this is, it's a European species of honeybee, and it is the most commonly found species worldwide. And honeybees are important because they provide more than 80% of the world's pollination. Now, how the process of pollination works is the honeybee will go from plant to plant collecting nectar, which is its food source and also what is used to produce honey. During this process, some pollen will collect on the hairs of its body. And while it continues this process, some pollen will drop on the plants that need it in order to reproduce for future generations. So, this is important for humans and other animals because we are highly dependent on this species for agricultural production. Without this species, our survival would significantly decrease. Colony collapse disorder, or CCD. What this is the phenomenon that of the decline of Apis mellifera throughout the years. So, 2 decades ago, many researchers began to notice that hives of honeybees were empty with no traces of a honeybee to be found, but their entire food sources were left behind. So, this began to cause question to where the honeybees were going or what was happening to them. So, throughout the years, it has been researched, and they have believed that colony collapse disorder is linked to some disorders that have been killing the bees. And some probable factors that result in colony collapse disorder are thought to be climate change due to changing temperatures, pathogens, and parasites. The lower right image is an image of a honeybee, and on its surface there is a red spot. But it's an ectosymbiotic parasite, which means that it lives on the surface of its host. This is known as a varroa destructor. What this does to the Apis mellifera is it targets its gut health, and it can be lethal. And then the upper right image shows Nosema ceranae, which is what we'll be focused on in this study. Nosema ceranae is a fungus, and it's a unicellular pathogen. What this does is it targets the gut epithelial tissue of Apis mellifera, and it can result in malnutrition, a shorter lifespan, and eventually death. The study is going to focus on the exposure of insecticides to Apis mellifera. And it is important to note that in a regular agricultural setting, that there are pesticides that are sprayed, and these include insecticides, fungicides, and herbicides. However, there has not been much research on the effects of herbicides and fungicides to honeybees because they are thought to be less lethal than insecticides. Previous research has also found that there could be correlation between pesticide exposure and the increase of vulnerability of pathogens such as Nosema ceranae. But it is also thought that pesticide exposure and the effects are highly dependent on which type of pesticide is used and what dosage amount is administered to the bees. So Apis mellifera do have an immune complex that has evolved throughout the years and become stronger. However, Nosema serenate has become stronger as well throughout the years, which is why it continues to be a threat even today. And in previous research, it has been shown that gene expression has been changed when exposed to Nosema serenate spores. And gene expression is important because it allows us to see if the exposure of certain insecticides or Nosema serenate spores can change the genetic makeup of these organisms. Here's an image that shows how the pesticides are sprayed in an agricultural setting and how they can get directly to the colonies and indirectly to the colonies. So first, the red box represents insecticides, herbicides, and fungicides sprayed. Some of those will be absorbed by the soil, and then they will continue to be absorbed by, in this case, the sunflower by its roots. And then some of these will then go directly to the colonies through soil drift and water contamination. And then on the surface level, the foliar spray will get to the surface of the sunflowers. And then some of that could directly go to the colonies through pesticidal drift. Those two, the surface and the underground, those are direct paths that the pesticide could get to the colonies. And then an indirect pathway is through pollination. So when the honeybee is going to collect nectar and pollen from this case, sunflower, the pollen and the nectar would already be contaminated by the insecticides. So when the bee goes back to its colony, the pollen and nectar that will be given to the colony will be affected, and this could have negative damages to the entire colony. Just for a bigger picture, not just one honeybee is going to be pollinating, it's going to be hundreds of bees in each colony. So this increases the exposure and the effects of these pesticides rapidly. So the study I'm going to be talking about was conducted by Al Najar and Bayer at the University of California in Riverside. This study was carried out beginning in August and ending in October of 2018. And this study was published by Nature Publishing Group on December 24th, 2019. Upon this study, Al Najar and Bayer had a couple of things in mind. So they believed that a larger quantity of Nosema serenate spores would be found in colonies that had been exposed to the insecticide rather than those that had been exposed to the control. They also thought that exposure to the insecticides in Apis mellifera would make them more prone to obtaining disorders that link to colony collapse disorder. And they believe that the gene expression of these organisms would change upon exposure to the insecticides and the Nosema serenate spores. So the way that Al Najar and Bayer approached the study, so they studied a total of 7 colonies and they divided them into 2 groups. One group would be exposed to the insecticide during their developmental stage as larvae, which can be seen in the image to the right. And then another group would be exposed to the insecticide during the early adult stage, which would be an example of the bee at the top. So in order to do this experiment, they used 2 different types of insecticides. One is the brand Sivanto, which is an a commercial grade insecticide that is used nationwide. And the reason why this insecticide in particular was selected is because it is advertised as a safeguard for beneficial insects such as Apis mellifera. And then flupyradifuron or flu, which is the active ingredient in Sivanto, was selected as a pure chemical substance that would be administered. The bees that would be exposed to the insecticide during their development would be used in order to observe if the effects of the exposure were long lasting and if they could be reversible in the future. So they studied a total of 7 colonies, and 3 of those colonies were exposed during their development, and 4 of those would be exposed during their early adulthood stage. The dosages of the insecticide that were administered were of 0.025 micrograms for the larvae and 0.645 micrograms for the early adult bees. These dosages of the insecticide were diluted in a double distilled water and then combined with a stock solution of sugar water containing 50% sucrose for a final concentration of 0.0043 micrograms per microliters. And the way that the solution was administered to the bees was through a sugar water feeder, which can be seen in the image to the right, where the honeybees go to the bottom of the feeder and they drink the sugar water solution. So the methods that were used, the bees that, in order to infect the bees with Nosema ceranae spores after the exposure period of the insecticide, at the before the beginning of the study, 100 working bees with a known Nosema ceranae spore infection were dissected and their midgut tissue was studied, where they obtained the Nosema ceranae spores that would be then combined with a stock solution of sugar water and then fed to the bees in the same way as they were the insecticide. And this exposure was done for 6 days, but they were absorbed for a total of 10 days. The last 4 days, there was no exposure to the Nosema ceranae spores, and this allowed them to test the intensities of the infection of Nosema ceranae and the exposure of the insecticide. So out of the surviving honeybees, 3 were randomly selected in order to quantify the effects. And these 3 were also studied in order to see if their gene expression changed. And using a real-time quantitative polymerase chain reaction, it measured the exposure of immune and detoxification genes and how they changed after the exposure to these 2 stressors. So what the results found, this is a figure of a plot survival. The upper image shows the bees that were exposed to the insecticide during their development. The x-axis represents each development stage, beginning at the third instar and ending at the emergence, and the y-axis represents the survival percentage. Each color correlates the control and the 2 insecticides, the red for the flupyradifuron and the blue for the Sivanto. The dotted line shows the end of the exposure period. And what is important to note in this figure is that the survival rate significantly decreased for the flupyradifuron beginning at the third instar. You can see the distinct line separation between the control and the Sivanto and the flupyradifuron. And even after the exposure period had ended, the effects were still there for the flupyradifuron exposure, and the survival rate continuously decreased. For the bottom figure, this shows the bees that were exposed to the insecticide during the early adult stage, and the x-axis represents the 10 total days that they were observed. The 6 days represented by the dotted line of where the exposure period ended, and the 4 days that they observed the lasting effects. The y-axis represents the survival percentage, and each color correlates to either the control, the flupyradifuron, or the Sivanto. And what is neat about this is that in contrast to the bees exposed during the development, the Sivanto formulation had a greater effect on the bees in their early adult stage, and their survival rate was significantly lower than those of the flupyradifuron and the control. But this figure particularly allows us to see that the survival rates do decrease much more upon insecticide exposure rather than just the control, which can't be seen as significantly for the developing bees as the line for the control and the Sivanto overlap. This figure shows a survival plot for bees that were exposed to the insecticide or the Nosema ceranae spores during the development, and then it also shows the bees that were exposed during the early adult stage to the insecticide and then to the Nosema ceranae spores. The line I want to focus on in figure A is the pink line, which the arrow is pointing to, and the circulation of Sivanto and Nosema ceranae spores. This combination showed to have the lowest survival percentage among all of them for the bees exposed to the insecticide during their development. And then for figure B, this shows the bees that were exposed to the insecticide during their early adult stage. The y-axis represents the set number of days, and the x-axis represents the percent survival. The line I want to focus on in this survival plot is the red line the arrow is pointing to, and the Savanto that it is circled. So what this shows is that the Savanto had a significantly lower percentage survival, regardless of if it was combined with the Nosema serenade spores. So all of the other combinations of the control and the spores and the fluopyridopherion and the spores were almost layering up on each other, but the Savanto shows a significantly lower percentage survival. And this also shows that the effects of the insecticide are greater for the bees in the early adult stage rather than the developing bees, where the effects are greater of Nosema serenade exposure. This figure shows the intensities among the two treatments of Nosema serenade spores and the insecticide. So figure A, and the x-axis that shows the 3 colonies that were exposed to the insecticide during their development, and the y-axis shows the Nosema spore count per bee with the mean plus the standard error margin. Colony 1 showed a greater amount of Nosema serenade spores for the bees that were exposed to the fluopyridopherion during their development. And across the board, the other bees did not have as much significant change in the amount of Nosema serenade spores found. For figure B, this shows the bees that were exposed to the insecticide during the early adult stage, and then later on exposed to the Nosema serenade spores. Colony 5 in particular had the greatest count of Nosema serenade spores, and this was higher for the bees that had been exposed to the fluopyridopherion first, and then closely following to the Savanto. This figure showed the bees that were exposed to the insecticide during their developmental stage, and it shows the gene expression for 2 detoxification genes of CYPD1 and CYPQ3. The gene expression was seen greater for the CYPD1 gene for the bees that had been infected in the control and the fluopyridopherion, and it was higher for the non-infected bees for the Savanto exposure. For CYPQ3, the abundance of this gene expression was higher for bees that had been exposed to the insecticide when they were not infected with the Nosema serenade spores, and it was higher for the infected bees when they had been exposed to the insecticide control. This figure shows the bees that were exposed to the insecticide during their early adulthood stage and then exposed to either a control or the Nosema serenade spores. What is shown in this figure on the x-axis represents the control and the 2 insecticides, and the y-axis represents the abundance of transcription of each gene expression. And for the chitinase 5, there is a significant increase in the gene expression for the bees that were infected, regardless of which insecticide they were exposed to. And for the defensin 1, the gene expression was higher for the non-infected bees, regardless of which insecticide they were exposed to. So what this brings us to the conclusion, what Al-Najjar and Bayer found was that insecticide exposure could be a potential contributing factor to colony collapse disorder, as the survival rates did decrease significantly upon exposure to these stressors. They also saw that the bees that were exposed during the development, the effects of the exposure did have long-lasting effects, and that they were not able to be reversible. They also were able to see that the bees that were exposed to both the insecticide and the spores during the development had a lower survival rate. So this showed that Nosema serenade had a higher effect on developing bees rather than those exposed during the early adulthood stage, where the survival rate was significantly lower to those that had just been exposed to the formulation of the insecticide. And the infection intensities were seen to be higher in colonies that had been exposed to both stressors of the spores and the pesticide, rather than those that had been exposed to one or the other or both controls. Overall, what this study means for the future of honeybees is it allows us to better understand the pesticide tolerance of these species, and it could create better breeding techniques in order to give them a better coping mechanism for sublethal dosages of pesticides. It also brings awareness for the agricultural setting in order to either use sublethal dosages of this pesticide or to find alternatives for pesticide use. And that is the end of my presentation. Thank you for listening.